So good morning. I would like to say I feel like Madonna before a show. Thank you very much for the invitation. I really came out from Brazil for this meeting. And I would like to thank especially Babette and Suzanne for this nice invitation. It's for me a great opportunity also to talk with the local groups about a possible interaction. So what I will show is more or less what we are doing in Brazil and what we can think about our work together. Yeah. So I will talk especially about transcriptomics of plant response. We are doing some works with native and cultivated plants against environmental stresses. Yeah. So as was explained at the previous talk, plants also get stressed, not only us. Yeah. And these stresses may be biotic. Yeah. They include many different microorganisms and also insects, nematodes, and abiotic. Yeah. In the case of Brazil, we are very concerned about these three ones. Yeah. Insects is not a large problem because they can be easier controlled. And in my region, especially drought and salinity, because in northeastern Brazil, where I live in Recife, we have a large dry land area. So the question we have is how long plants take to perceive such stresses and how do they respond to such stresses, looking at transcriptomics. Yeah. So since the 50s, people know that plants can respond quite fast. Also this last talk was an example of that. And this is shown by the volatile compounds. Yeah? So that plants, they perceive and they can send some chemical signs that show not only they have perceived, but also they warn other plants, and all plants may react because a single plant was attacked. In the case of biotic stress, yeah, especially the microorganisms, they can synthesize molecules that are called AVR genes. And these gene products, they are perceived by plants by resistance genes that activate this gene-to-gene interaction cascade that also will induce other systemic acquired resistance, PR genes, including peptides in this case, and also other hormones and so on, and transcription factors, and also volatile compounds. For drought, it's very interesting because they are more complex than biotic stress. It is interesting because very near related species may have completely different reactions. And in the case of some plants, you have the same species or very nearly related that survives as a very severe drought or still salinity, and another one that doesn't survive. So this may be cleared maybe by gene evolution and adaptive evolution. And many have said this is a multifactorial polygenic inheritance and a complex physiological process. So what we have been seeing in this case, that we have two kinds of responses. These early responses that include transcription factors, ABA biosynthesis, yeah, and signaling molecules like kinases and phosphatases, or later responses that include protective proteins, water channels, proteases, osmoprotectants, and detoxifying enzymes. Yeah. And we have been looking more nearly to kinases, transcription factors, these water channels, yeah, that includes mainly aquaporins, yeah, osmoprotectants, and also genes we have a known function or they have never been described, what we call no hits. Very early it was observed in Arabidopsis when they evaluated 7,000 possible genes that may be active in abiotic stress, that cold, salinity, and drought have many genes in common, especially drought and salinity. And we have been focusing in these two kinds of abiotic stress. These genes that are represented here are only the genes that have been induced, have a fold change higher than 2.5 times. Also in Arabidopsis, it's clear that this response is quite fast, yeah? In the first 2 hours, most of the genes are activated and they have activation level until 5 hours in the case of salinity, and until 10, and then this is decreasing and the second group of genes is activating later on. I'm showing this because for most plants, transcriptomics has been done later than that, yeah? They have started collecting data with more than 10 hours. So many studies don't look these first hours. This is starting in the last 5 years only. So the projects we are running are the CALPI Genome Consortium. This is this CALPI, yeah, this Kubone in German. It's not very popular here, but it's very, very important for Brazilian Northeastern region, for the Middle American region, and especially in Africa. It's the only bean that is growing in Northeastern and Northern Brazil, because other beans, they don't survive the environmental conditions. This is our network, and we are here in Pernambuco, and this is, I coordinate this network. So we have done especially expression profiling with within this project, and we have generated many different libraries, considering especially salinity and drought using roots, virus resistance against two very important viruses in Brazil, and also leaf injury, because we have to injure the leaves for inoculating the virus. And here we have also some abiotic stress, yeah. So what we have seen is that more or less of 18% of the genes in CALPI are unique. That means no other plants, is no hits by other plants. And this shows how less erudite this genome is not so breeded like other legumes. The other project is the soybean genome. We have just released a special volume in this journal. And I'm again here, the only group in Northeast, because soybean is more important in this Brazilian area. Yeah. And I coordinated this special volume, but the coordinator of the project is here in Paraná, in southern Brazil. So in soybean, we did also focus on stress-induced genes, especially water deficit and Asian rust. Yeah? I'm not talking about these microRNAs. I will talk more about subtractive, supersage, and RNA-seq libraries. And what we do with all this data, yeah, we do a very basic analysis to annotate this data and have it in a data bank. So mainly including the identification of more expressed genes, comparing the most interesting situations, for example, the tolerant stressed against the susceptible stressed or against the control, but we compare more or less all libraries against all libraries, yeah. And then we do an overall categorization, what we call primary annotation. And then we look about the structural features and molecular roles. We also do some analysis of specific gene families. Yeah? I'm not going to talk about each step here, but what is really important is that we find differentially expressed tags or reads, and also unique genes that appear in one library, for example, the resistant one against a virus, and is absent in the other libraries, yeah, after this stress, application of this stress. And also when we do that, we do some bench work, and also we are very focused about gene evolution. So I will show some data. It's are very large projects, but I selected some slides. This is, for example, the results of the GO categorization under salinity stress, yeah, for CalP. And just after these first 3 times that were bulked to form a single library. So if you look, you see, as expected, a lot of gene categories that are known for abiotic stress, but also some that are known for biotic stress. And this is also very often because we know there is some crosstalk between both types of stress, and also some that are responding to both, inclusive oxidative stress that was the focus of the previous talk. When we look at this distribution in the cell, yeah, cell compartments, what we see is that first the controls, they are ever, the external and susceptible controls, they are ever lower in this, only considering the most expressed genes. They have a fold change of 2.5 times and more, yeah. So they are less expressed, yeah, than the normal control, than the tolerant control. That means these tolerants, they start in this stress already with some genes active that are absent in the susceptible control. Except here in this case, yeah, in the extracellular region and extracellular matrix, yeah. And for the others, they are normally higher induced, and this is really dramatic here in the tolerant after 2 hours in these 2 cases, the extracellular region and the extracellular matrix, where there were almost no expression before the stress. So we have been then looking at these genes family by family. This is an example of the kinase, in this case after mosaic virus induction and salinity. Yeah. What we saw is that we have 901 differentially expressed kinases in 11 classes. Yeah. And when we look in the tolerance, we have ever unique kinases expressed that are absent in the susceptibles. This was already published, yeah. So we have a special interest in signal transduction, especially transcription factors. So here I'm showing after this time, yeah, how many differentially expressed transcription factors we have identified in CalP, also some also DREB and F, yeah. And some of them, they are co-expressed, yeah? That means they appear in both susceptible and resistance, but they are more expressed in the susceptible, yeah? Up to 20 times or more. And then we have also these unknown proteins. Yeah? I have a very high interest to identify their function. We don't have this kind of study you have here. This is why I want to collaborate. We are not still transforming plants, making knockdown and overexpression. And I think some of these, some have 70-fold more expression in the drought tolerant than in the drought susceptible. So they may be very interesting genes, yeah. So in this case of cowpea for salinity, we have 234 of such genes, yeah. So this is in the case of mosaic virus, yeah. In this case, we have also a higher induction after stress. This is the first minutes. This is after 16 hours, yeah. And here is the negative control. Some are in common, they are not so interesting, but these exclusive genes, they are very interesting. And among these, especially this group, we have 375 with no hits. They have no similar genes in the data banks, yeah. No, my time is okay, yeah. So in this case here, I'm only showing for super sage. This is one of the expression methods we have used. After virus inoculation, also here, the same than in salinity, yeah? A very strong induction in the extracellular region and extracellular matrix, yeah? Well, for dehydration stress, this means in soybean, we didn't do cultivation in pots, but we did hydroponics because soybean is very sensitive against drought. And then we did this stress that was for very short period of time, from 30 minutes until more or less 5 hours, each half an hour. And then we have chosen these two responses, response to hormones and response to water deprivation, considering gene ontology categories, yeah. And we selected the genes that were in this class, some of them, to make qRT-PCR, for example, this one, yeah. But we have here many candidates that we are doing that, yeah. And some of them didn't show so high expression by qRT-PCR as expected, but other they were really nice, yeah, like this Erebia. That is also a nice candidate for genetic transformation and transient expression, yeah. So, but in all cases, tolerant were higher expressed than susceptible, yeah. And in some cases, the susceptible, they had a very lower expression than the tolerant, yeah, more than we expected. So I will talk very shortly about a future perspective we have in the plant defense exome project. This is something we have been working on, mainly on bioinformatics also. And what we are doing, we are we have selected a highly curated seed sequence bank, and regarding transcription factors, antimicrobial peptides, that means defensins, cyclotides, snakins. They are most of them are the category PR14. Yeah. And R genes, but in this case, the non-TIR and TIR NBS LRR category. And we have collected data from 63 plant species that are available in data banks from these two families, yeah? And also model plants, yeah? Especially Arabidopsis and rice. Yeah? So here is only to show the categories of R genes, yeah? And what we are focusing, as I said, was this TIR and BS-LRR, yeah? And then this non-TIR. So the work includes the capture of the sequences using the seed sequences, and then they are aligned. Then we do this T-BLAST-N to identify conserved domains, yeah? We do a protein matrix alignment, and then we have a secondary data bank, yeah, with these plant defense candidates. This is for defense. We have another for transcription factors. And then again, multiple alignments, and then you use this hammer program because some of these genes, they cannot be identified with normal alignments, yeah. And then we do the similarity tree only to group them into categories that will be used for further study. And then this maturated database, this is more or less ready, yeah. This shall be used for anchoring against a reference genome. This is the next step. For legume, we are using Phaseolus vulgaris, yeah, the common bean. Yeah. And then we do this definition of gene architecture and primer design. And we have then in-target control, conserved exon, and out-target negative control. The next step shall be this wet lab, yeah, work. So the last part, yeah, wet lab, with this work, I'm short of time, we are able to capture these genes from any plant group, yeah. And this is what we intend to do using plants that are native plants of Brazil, and they are good candidates for giving interesting new genes, yeah. So concluding, dramatic changes occur after stress perception, early expression of transcription factors and signal transduction genes, induction of many unknown genes or no hits. We intend to do, yeah, time frame evaluations, also in known models, yeah, they are really necessary. Expression validation and report gene trials is something we are willing to do through partnerships, yeah. And for some genes, for some, especially these no hits, I think systems biology, pathway reconstruction would be a nice idea, yeah. So I have been talking with Adriano that we'll talk later on today. He works with bioinformatics. This is something very promising, yeah. And comparative exomes for giving gene families, yeah, because this is very cheaper and faster than transcriptomics, yeah. So I only say these are some of our candidates for bioprospection using this exomics, yeah. This is my work group in Brazil. You are all invited and very welcome if you come to Recife. It's a very nice place, I say. The colleagues have been there and they like it. I hope they come back, yeah. And these are important partners, yeah, Professor Kido, Sergio Crovella. This is a postdoc, yeah, Valeska. This is a German partner aus Frankfurt, Professor Ginta Karl. These are bioinformaticians from São Paulo, and these are all my students, yeah. So thank you very much.